Good day everyone! In this video lesson, we're going to tackle about enzymes. Human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Anselme Payen, was the first to discover the vital force that drove these reactions and name it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up the biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action easier said. But how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for a reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily.
For two molecules to react, they must collide with one another. They must collide in the right direction or orientation and with sufficient energy. Sufficient energy means that between them, they have enough energy to overcome the energy barrier to reaction. This is called the activation energy. Enzymes have an active site. This is part of the molecule that has just the right shape and functional groups to bind to one of the reacting molecules. The reacting molecule that binds to the enzyme is called the substrate. An enzyme catalyzed reaction takes a different route. The enzyme and substrate form a reaction intermediate. Its formation has a lower activation energy than the reaction between the reactants without the catalyst.
This graph illustrates how enzymes speed up chemical reactions. Remember that in a chemical reaction, the reactants interact to form a product. The chemical reactants for these examples are on the left and the products are on the right. The wall that separates them represents the activation energy. You can think of this as an energy speed bump. The larger the bump, the slower the reaction. The yellow speed bump represents as a chemical reaction without an enzyme, and the orange speed bump represents the same reaction with an enzyme. As you can see, the orange speed bump is a lot lower. This is because the enzyme acts to physically bring the reactants together. By doing so, it increases the efficiency of the reaction and lowers the amount of energy needed for the reaction to occur. Since less energy is required, the reaction occurs at a faster rate. Notice that the enzyme does not influence the energy level of the reactants or the products, but only the amount of energy is required during the process of the chemical reaction. Without the use of the enzymes, many of our body's processes, such as digestion and the processing of nerve impulses, would simply occur too slowly. The addition of enzymes, however, speeds up these chemical reactions, making life as we know it possible.
Lock and key theory. Lock and key theory of enzyme, also called Fisher's theory, is one of the two models that describes enzyme-substrate interaction. The lock and key model states that the shape of an active site of an enzyme is exactly complementary to the shape of the substrate. The substrate is like a key that fits into an enzyme site, like a lock and a key, only the correct size and shape of the key would fit in the hole of the lock. And the same way, only the correct size and shape substrate would fit in the active site of an enzyme. The lock and key model shows specificity of an enzyme for a particular substrate.
Let's take a look at an example. One example of a substrate is starch, and the enzyme that it binds to is called amylase. So if they come into contact with each other, the starch will bind to the active site of the amylase. Amylase catalyzes the reaction, which means it speeds it up. And in this case, the reaction taking place is the breakdown of the starch into its product, and starch breaks down into glucose. Here we can see there are two glucose molecules. Those are products. Different enzymes have different sizes and shapes because their amino acid chains have been folded differently, giving them a different 3D structure.
Now let's proceed to induced fit theory. This theory by Daniel Kochland in 1958 was introduced because of the drawbacks of the lock and key model
as they realized that this model was not correct.
The lock and key model assumed that the active site of the enzyme has the same conformation as that of substrate. The same conformation of the active site makes the enzyme very specific for the substrate. The lock and key model also assumes that the active site is rigid and have the same shape as that of the substrate, when it is actually flexible and can have different conformation. This model assumed that the amino acids present at the active site will determine the specific binding of the substrate with the enzyme. Once the substrate binds the enzyme, the conformation of active site gradually changes and attains the shape of the substrate. Hence, it is known as induced fit theory or model. According to this theory, whenever a substrate binds to an enzyme, it induces conformational change to the enzyme, which strengthens its binding. The induced fit theory proposes that enzymes are not static but can alter their shape upon substrate binding. This dynamic interaction optimizes the fit between the enzyme and substrate.
Here, we can see a visual representation of how the enzyme, which is depicted in red, changes its shape to accommodate the substrate depicted in green and violet. Substrate binds to the enzyme's active site, and as this substrate binds, the shape of the active site changes slightly, allowing enzyme-substrate complex to form. If the substrate enables the active site's shape to change in the right way, then reaction takes place and an enzyme product complex is formed and products are then released from the active site.
Factors influencing enzyme action. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include: number 1, temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes.
pH. Amino acid present in active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrate to bind. Extreme pH values can denature enzymes.
Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be available to bind to substrates. However, after a certain concentration, any increase will have no effect on the rate of the reaction.
Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration.
Inhibition of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzyme in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors: the competitive inhibitors and the non-competitive inhibitors. Competitive inhibitors occupy the active site and prevent a substrate molecule from binding to the enzyme, while non-competitive inhibitors attach parts of the enzyme other than the active site to distort the shape of the enzyme.
As we conclude our journey into the realm of enzymes, it's clear that these tiny molecules play an immense role in shaping life as we know it. From aiding digestion to powering cellular processes, enzymes are the silent architects of biological functionality. I hope this lesson about enzymes has sparked your curiosity. So that's all for today, and thank you for watching this video.